All right, welcome. Thank you all for being here and for joining us. My name is Danielle Forchetti. I'm an educator working with the Western Pennsylvania Conservancy, which is an organization that works to protect and restore forests, watersheds, wildlife, natural areas. We also create green spaces and gardens, and we preserve Falling Water, which is a UNESCO World Heritage Site and a masterpiece of architecture in Western Pennsylvania. I'll allow my co-facilitator to introduce himself now.
Hi, my name is Patty Greist, and I'm a gifted support teacher at Seneca Valley and in the high school. And I work with students in 9th through 12th grade. And we're working today with students that are studying with us in our Gene Text program.
So we are joined by a panel of scientists who work both in the field and in the lab and are contributing to our understanding of the health of our rivers and streams, as well as some of the really interesting critters who live there.
My name is Eric Chapman, Director of Aquatic Science for the Watershed Conservation Program based out of Indiana, Pennsylvania. I've been with the WPC since 2006, and we've been working with hellbenders, freshwater mussels, brook trout, crayfish since that time, and really got involved pretty heavily with eDNA in 2014. I'm really excited about the collaboration with Seneca Valley High School. I think it's an amazing opportunity that you guys have.
So let's go ahead and just open the floor for a discussion. Mark, would you like to get us started with a question for Eric or for the group, just something to kick off our discussion?
So I was wondering how exactly we would kind of build on what they did and do that effectively and make like our own kind of experiment out of that.
Yeah, I think this summer, well, not this summer, this fall is when we collect our eDNA samples. And as a result of this partnership, I'm going to carve some time out in September to go out and sample a whole bunch of new streams using eDNA. So I wasn't going to do that, but since this is moving along at the pace that it is, I want to go out and sample a whole bunch of places and send you guys all of the resultant from that eDNA filtering that we'll do. We'll send you guys the samples and have you guys work that information up for us. So there's a bunch of new streams that I want to look at that we've never surveyed before. So that'll be really cool. And, you guys could take that filter paper and isolate the hellbender DNA, but there's also people that, want to take pieces of it and, look at the freshwater mussel community that might be present. There's a limitless opportunities with the work that we'll do this fall for you guys.
Just realized no one ever officially defined eDNA. So if someone wants to, I can do it. So the listeners know what we're kind of talking about when we say eDNA studies.
Sounds like a great thing for you.
All right. So when we're talking eDNA, we're talking environmental DNA. It's out there in the environment. It's almost always from bodies of water where you collect. There's this ambient just DNA floating in there from all the life forms inside. Whatever cells brush off, and now there's just the DNA in there. So then what we do in eDNA studies, we collect these water samples, and then we filter them down just so it's just all the DNA from all the animals and plants and whatnot in the stream. And then we use special primers which attach onto the DNA for specific targeted genes, the specific nucleic acid sequence. So then whenever those latch on, you have a match. And if you have a match, then that means there is in fact whatever organism you're looking for in that body of water.
I wanted to build off of what Ty said about the primers. So specifically for Sydney and I, and for all other eDNA researchers, it's really important to make sure that your primers will not cross-reference with another species in the region you are looking for. Otherwise, this could give you a false positive reading. So that actually requires a lot of research into making sure you know what kinds of organisms are in the waterways you will be looking at.
We have had, in the beginning, not so much as of late, we used to get a lot of false positives. So we would take an eDNA sample that was run at our lab that we work with, and they'd give us back, hey, you got a positive. And so I'd take my crew and we'd go out and sample, and we'd lift 1,600 meters of stream, lift every single rock for 11 hours straight, and could never find a hellbender. And it was really frustrating for me from the research side, because the genetics people were supposed to tell me where to go work, and then I'd go and I wouldn't find anything, and it was frustrating. But I think that's part of eDNA. Was when it first started for us in 2013 and 14, there was a lot of learning. We suffered from false positive. We suffered from cross contamination, which I don't know if you guys have seen in the lab, and that can be really, really frustrating. But that's all part of learning. Scientific exploration and learning is working through those issues that you come up to. And there's no better project in my eyes for student work than eDNA.
To your point about the contamination, I think we did have a problem with that last year. So it is definitely a whole learning process. And this year for Sydney and I, we're dealing with a problem with our own primer. We are experiencing something called primer dimer, where the primers are bonding to each other, and that can make the accuracy of our quantification not as well. So that is something we are going after too, and it is just a learning process of trial and error. So yeah.
But you guys are learning it. That's the most important thing. It's so cool.
Yeah, we've also had issues with false positives in our eDNA research. For most of the year, we've been getting false positives or strange things with our controls, and only recently have we been able to actually get an accurate, good-looking data in our results.
Yeah, there's nothing more frustrating than getting a positive in your negative control. But for people that don't know anything about eDNA, when you guys want to explain, what a false positive is with a negative control, maybe?
Control would be a sample that does not contain any of the DNA that you're looking for. And if you get a false positive, that means that the machine is detecting DNA that is not actually there.
Which is very, very bad.
Yeah.
Which I guess sends you out into streams looking for things that aren't there.
Yeah, it could. Or you sit there and scratch your head and go, did I contaminate the sample when I collected it? Did you guys contaminate it in the lab? Probably my funniest story ever, I told you guys when we were doing the run through, was the false positives that we had with the Smithsonian National Zoo when we were doing my very first eDNA project. We were getting false positives with our negative controls like crazy. And we were able to work it back to the water that we were using was not distilled water for the rinsing. It was it was just spring water, and it was pulled from a spring that actually had hellbenders in it. So it contaminated everything, and we lost like a whole year's worth of work. So it was very frustrating.
And you guys have gotten to learn from those kind of mistakes, because you guys didn't use spring water at any point, I don't think, because we learned that from Eric that wasn't going to be functional.
No, we don't do that. And I openly admit that I am a field guy. I am not a lab guy. So I totally respect what you guys do and have a very elementary understanding of what happens in the lab, because I'll be honest with you, I've never been in an eDNA lab before. And whenever hopefully all of this gets past us, I'd love to come to the lab and, watch you guys run your stuff.
So when you're doing field work directly in the freshwater, like how you were saying that you're turning over rocks and looking for these organisms, how do you limit the invasiveness, or I guess make your environment sterile, or just techniques that you have to limit the invasiveness of an already endangered organism that you're looking for, as opposed to just taking the sample in eDNA, which is supposed to limit the disruption of their habitat? And can you kind of speak to the comparison of accuracy between macro and microscopic studies?
Yeah, for sure. So when we're doing eDNA, that is the most minimally invasive thing that we ever do. So you walk out and you're taking 2 liters on the left descending bank, 2 liters in the middle, and 2 liters on the right descending bank, and that is it. You're not disturbing. There's no touching of rocks. There's no substrate disruption. Because I don't want to get any suspended particles up in the water column, because then that means like 35 hours of filtration. We have learned that you don't ever sample after a rain event, because turbidity is not your friend. We've had samples where, we've filtered for 35 hours straight. So I'm very, very particular about when I go out and collect eDNA samples. The flip side of that is when we're actually out doing our surveys for hellbenders. The only way that you can truly quantify the population by using mark and recapture with pit tagging or things like that is actually lifting the rocks and catching the animals. It's been a big battle in the hellbender researcher community. Some people are totally against lifting and turning, and some people say that's the only way that you do the research. I'm kind of in the middle. I feel it's important to do the lift and turn surveys. We only will survey a given reach a stream once a year. And I'm very, very particular about putting rocks back in the exact same position where we found them. You also have to have that Pennsylvania Scientific Collector's Permit to be out doing the work. So, the general population is not out flipping 1,000-pound rocks looking for the animals. So I consider it's a trade-off. That's how a lot of things that we do, you have some things that are going to have a little bit of negative impacts, I guess you could say, but I feel it's part of the overall experience that you have to. I can't measure an animal and tell you whether it's male or female by just looking at your eDNA signature. You can tell me, yes, they're there, but we can't get any idea of what the population looks like. And to me, that's really important. We didn't know if these animals are actively reproducing. Are there juveniles present? Is it a geriatric population where there's only adults? That's concerning. If there's only adults, they're not reproducing. That population's probably going to wink out, in the next 50 years. So that's a really good question, Sydney.
What did it take, for you to be able to get the job you have now, or the people you work with? What steps did you have to take and whatnot?
So I started out and did my undergrad at Indiana University of Pennsylvania, way back when. Got my bachelor's degree there in biology, and then moved to New York City. So I started my graduate degree at Queens College in New York City, and was working full-time as a research scientist there. And then I worked for the Dean of the College of Science and Math. And then we moved over to New Jersey to Montclair State University, where I worked another 5 years as a research scientist, lab manager, and then finished my master's degree there, and then came back to Western PA in 2006. And I think that the academic side, the research knowledge and experience that I had worked really well for me to get into WPC. You guys are light years above where I was when I started. I can't even imagine the experiences that you guys have already that I didn't get until I was 20 plus years old. I think it's fantastic what you guys are doing right now. You guys are answering questions, research questions, while you're 9th, 10th, 11th, 12th graders. To me, that's amazing. Working at WPC has kind of been a balancing act of going out and doing all the field work, but still, having to come and write grants and do all the, the administrative side of things that make sure that we're able to go out into the field to do the work. So yeah, being able to work together in a team, which you guys are already doing, you can't just take on the world by yourself. So you have teamwork side of things. It's really important.
What are some things to be thinking about building skills that can help contribute to this pathway?
I would say, if people are out and about, in their streams, if they're out, fishing with their grandfather, or, walking on their property, and they, see something that they don't recognize. They see a, Jesus, 2-foot long salamander here. I've never seen one before, and they take a picture of it, because you guys all have your phones with you all the time, because I have 2 kids that are your ages, and you could send that to me, or you send that to WPC. That's a way of kind of looking at what's out in the real world, and I don't know what this is, but there's people that can tell you what it is, and it gives us a new location that we could go out, and do an eDNA survey, or go out and do a regular lift and turn survey. I think that's one way, just being out in the landscape, and observing what you see in nature is really important.
I think from the science perspective, just kind of learning about how to create an experiment and formulate questions, maybe contacting your teachers or your science teachers, because that's how our 2 science teachers that we talk to the most in the lab aren't even our current teachers right now, but we had them at some point. So definitely just keeping in contact with the STEM related teachers that will give you a lot of guidance.
Yeah, that's a great point.
Well, I want to thank everyone here for contributing to this discussion. Again, we could probably go for another 10, 15 minutes and not even scratch the surface. So thank you all so much for the work you're doing to protect and restore our rivers and streams, and all the work you're doing to study these fascinating species that make those places their home. All right, thanks so much. Take care.